What is cleaning validation? Imagine baking a chocolate cake, then a vanilla cake in the same pan. You'd want to wash it first, right? In medicine, this is critical. Cleaning validation proves, with real evidence, that equipment is cleaned so thoroughly that no residue from one medicine contaminates the next. It's not just about looking clean, it's about being scientifically, verifiably clean. Health authorities like the FDA require it because cross-contamination can be dangerous. Even a tiny amount of the wrong drug could harm a patient. Cleaning validation is our safety net, ensuring every batch of medicine is pure and safe. It's a structured, documented process that guarantees equipment is ready for the next product. This process protects patients and ensures the medicine works as intended. Contaminated medicine could be less effective or cause unexpected side effects. By validating cleaning, we guarantee the integrity and quality of every dose. It's a promise. What's on the label is what's in the bottle, nothing else. Cleaning validation is a cornerstone of good manufacturing practices, or GMP. It involves careful planning, precise execution, and meticulous documentation. Every step, from developing procedures to testing results, is recorded to create a paper trail. Validation isn't a one-time event. It's repeated to ensure ongoing effectiveness. It's about being proactive with patient health. This is why cleaning validation is a big deal in pharmaceuticals. Consider it. Can't we just wash equipment until it looks clean? In science, looks clean isn't enough. We need to measure cleanliness, turning a subjective idea into an objective scientific goal. Calculations set a safe, measurable limit for how much residue can remain. These limits are based on toxicology, ensuring even the most sensitive patients are protected. Math makes safety measurable and consistent across the industry. Standardized calculations mean medicines are equally safe, no matter where they're made. This creates a universal language of clean in numbers. Inspectors want to see data, not just hear, "We cleaned it well." Calculations are the foundation of our evidence. They show we understand the risks and have a scientific basis for our cleaning limits. It's not enough to say we did a good job. We have to prove it. These numbers are our proof. They demonstrate our cleaning processes are robust and effective. That's how we keep patients safe. The first big calculation is the permitted daily exposure, or PDE. PDE is the maximum amount of a substance a person can safely be exposed to every day for life. Toxicologists determine this by finding the highest dose with no negative effects, then dividing by safety factors. The formula PDE equals sign NOAEL x body weight, F1 x F2 x F3 x F4 x F5. NOAEL is the no observed adverse effect level. Safety factors account for differences between animals and humans and among people. These factors make the PDE very small and extra safe. For example, if NOAEL is 10 mg/kg/day, body weight is 50 kilograms, and safety factors total 100 PDE equals sign 5 mg day. This means a person could be exposed to 5 milligrams of the substance daily for life with no harm. PDE is the foundation for all other cleaning safety calculations. It's a health-based limit focused on patient safety. Once we know the PDE, we can set acceptable residue limits for equipment. The PDE puts patient well-being first. That's science protecting health. With our PDE, we calculate the maximum allowable carryover, or MACO. MACO translates the patient-safe dose into a total residue limit for all equipment. The formula MACO equals sign PDE x minimum batch, size of product B, maximum daily dose of product B. For example, if PDE is 5 milligrams, batch size is 100,000 G, and max daily dose is 200 G, MACO equal sign 2,500 milligrams. This means all equipment combined must have less than 2,500 milligrams of residue. Scientists test equipment to ensure residue is below this limit. MACO is the practical, measurable cleaning target. It connects toxicology data to real-world manufacturing. MACO ensures cleaning is both achievable and safe. It's the number we test against after every cleaning. This calculation bridges patient safety and manufacturing reality. MACO is critical for safe, consistent medicine. That's our ultimate cleanliness limit. How do we measure tiny residues? One popular method is total organic carbon, or TOC. TOC doesn't look for the specific drug. It detects carbon, the backbone of most drug molecules. The TOC analyzer measures total carbon in a sample, letting us estimate drug residue. First, we calculate what percentage of the drug's weight is carbon. For example, if compound X is 60% carbon, that's our conversion ratio. The formula residue amount equals sign TOC reading X sample volume carbon ratio. If a swab in 0.1 liters of water gives a TOC reading of 2 mgl and the drug is 60% carbon, residue equals sign 0.33 milligrams. TOC is fast, sensitive, and detects all organic residues, not just drugs, but also cleaning agents. Its strength is speed and sensitivity. Its weakness is non-specificity. High TOC could mean drug residue or something else. That's why TOC is often used with more specific tests like HPLC. TOC is a powerful tool in our cleaning validation toolbox. It helps us quickly check if equipment is truly clean. Combined with other methods, it ensures thorough validation. That's how we track down invisible contamination. All these calculations and tests require a solid plan. A cleaning validation program starts with a master plan outlining policies, responsibilities, equipment, and products. Next, we develop and validate the cleaning procedure, choosing agents, temperatures, and methods, then proving they work. Once the procedure is set, we write a validation protocol, a step-by-step guide for testing, including acceptance criteria like MACO. The protocol specifies how many successful cleanings are needed, usually 3 in a row. It details where and how to sample, and how to test those samples. After testing, we compile a final validation report. The report summarizes all data and compares results to our limits. If everything passes, the cleaning method is officially validated. This documentation is proof for inspectors and assurance for patients. A strong plan ensures every batch is made on clean, safe equipment. That's how we build trust in medicine. Where do we test for residue? We focus on the hardest to clean spots, worst-case locations. These are areas with complex geometry corners, gaskets, valves, or mixer blades. Flat surfaces are easy, but nooks and crannies can trap residue. Hard-to-reach places, like the tops of tanks or inside pipes, are prime candidates. Equipment material matters too. Plastic or glass may hold residue more than steel. Gravity plays a role. Residue can settle at the bottom of tanks or low points in pipes. We also consider the product. Stickier drugs need extra attention. By mapping out these challenging spots, we target our swabbing for the most meaningful data. If the toughest spots are clean, the rest likely are too. This approach saves time and ensures safety. It's about working smarter, not harder. Swabbing worst-case locations gives us confidence in our cleaning. That's how we ensure thorough validation. Let's connect the dots. PDE, MACO, TOC, and swabbing all work together to protect patients. It starts with the PDE, a health-based limit rooted in toxicology. PDE is translated into MACO, a practical residue limit for equipment. MACO gives the cleaning team a clear target. Swab samples and TOC analysis test the hardest to clean spots. Lab results show the actual residue. We calculate the total residue and compare it to MACO. If it's below the limit, we've proven the equipment is safe. This chain, from PDE to MACO to lab data, creates a traceable scientific argument for safety. Every step builds on the last, ensuring quality control. The process ends with a clean bill of health for the equipment. That's how we protect patients, every batch. After all the calculations, plans, and swabs, what have we achieved? We've built a scientific shield that protects patients. Every medicine you take is pure because of cleaning validation. It's a systematic promise, backed by data, that your medicine contains only what it should. Contamination can make drugs less effective or even dangerous. By mastering these concepts, manufacturers uphold the highest standards. Calculations turn complex science into a simple pass-fail test. Scientific rigor replaces guesswork with certainty. This process allows safe, life-saving drugs to be made in shared facilities. Cleaning validation is a quiet hero in pharmaceuticals. It's about control, care, and scientific excellence. Every person in the chain plays a vital role in public health. That's how we make medicine you can trust.